I now would like to share the presentation with academic Maurício Magalhães of the Nobel Prize winner, which has been generous enough to come to visit us, and we thank you very much for that. And I think the academic Maurício Magalhães will say a little something about your CV, which I think most everybody know about it. But essentially, I took the microphone to thank you for having decided to spend your time visiting us, and we are very honored to have you here. So again, thank you very much, Professor Richard Roberts.
Well, Professor Richard Roberts, Sir Richard Roberts, he's British from the beginning, but he lives in the United States for the last decades, and is the Chief of Scientific Officers at the New England Biolabs in Massachusetts. He received a PhD in organic chemistry in 1968 from Sheffield University, and then moved as a postdoctorate fellow to Harvard from 72 to 92. He worked at the Cold Spring Harbor Laboratory, eventually becoming assistant director for research under Dr. James Watson. He began work on the newly discovered type 2 restriction enzymes in 1972, and these enzymes have been a major research theme. Studies of transcription in adenovirus 2 led to the discovery of split genes and mRNA splicing in 1977, for which he received the Nobel Prize in Physiology or Medicine in 1993. During this sequence of the adenovirus 2, genomic computational tools became essential, and his laboratory pioneered the application of computers in this area. DNA methyltransferases as components of restriction modification systems are also of active interest, and the first crystal structure for the methyltransferase led to the discovery of base flipping. And he has a tremendous career scientifically and also as a human being. He's very much involved with the young scientists. This is why he accepted our invitation to come to Brazil, to be close to the scientists. And as I understood, probably you see, is now your main goal of your life to inspire the new generation for the future. Thank you very much, Professor, for coming. And for just, you know, he has just arrived today in the morning and will leave tomorrow. As you can see, the big effort he did to be here with you. Thank you very much, Professor.
Well, thanks very much for inviting me. What I'm going to try to do today is to give you a little autobiographical description that probably we could have skipped the introduction, to be honest, because I'm going to tell you a lot of the same things, but I will illustrate it with some things. But along the way, I'm going to try and tell you a few things that I learned during my career that I think can be very useful for students going forward. And there are a number of lessons that come from this, and I will tell you more about them as they come along. And then also tell you at the very end, summarize all of this. So I was born in the city of Derby, which is shown in the middle of this diagram on the left. This city was right on the path that the Germans were taking to try and bomb the Rolls-Royce factory, which was nearby, which was making all the jet engine, or the engines that were the RAF were using to bomb Germany. And they were not very accurate. And I lived on a street, and the bombers flew straight over that street. One side of the street got almost totally destroyed. Fortunately, it wasn't the one that I lived on. And so this was the first example of luck that happened to me in my life. It's only relatively recently, when I've been talking about the importance of luck, that it struck me that this was actually the very first bit of luck I had. Now, when I was 4 years old, my father, who was a motor mechanic, and my mother was a homemaker, they moved to the city of Bath in the southwest corner. You can see it circled on the right-hand diagram here. And I grew up in Bath. And that turned out also to be pretty lucky. Bath is a beautiful city. If ever you go to England, everybody goes to London. You want another city to visit, hop on a train. 2 hours later, you're in Bath. You can spend the day there and have a really wonderful time. It's a very nice city. When I was young, from age 7 to 11, I went to this junior school, St. Stephen's School, and something very important happened to me there. Actually, two things important happened. One of those things was that the headmaster used to give me little puzzles to do, mathematical puzzles to do at night. And I loved them, and I would take them in the morning and, either show him that I'd done it or ask for help. And it never occurred to me until sometime later that I was the only person he was doing this to. He didn't do it to anybody else. And he really stimulated in me a real interest in solving puzzles, loving mathematics, and realizing that one could use one's mind in many creative ways. His name was Mr. Brooks, and he died just about 6 months after I won the Nobel Prize. And so he heard that I had won, and he was rather happy about this, and I was happy too. This was really something very important. So that was a very good thing that came out of that school. The other thing that came out of that school was that when you were 11 in England, when I was growing up, you had to take a test called the 11 plus test to see whether you were going to go to a grammar school, which was sort of higher education and heading for university, or you would go to a technical school, which meant you were going to go and be a craftsman of some sort. And this was all based upon this 11 plus exam. And what happened was that I went to school one morning and the teacher said, "Oh, sit down, I've got some questions for you." And we were all given a few sheets of paper as I answered these questions, and we did. And it later turned out that this was the 11 plus exam. We had no knowledge ahead of time. We didn't have to SWOT for this exam. There was no pressure to try and figure out how you could beat the odds, if you like, when you take exams. And for me, I've always felt that was the way to take exams. I don't like this idea of spending weeks studying, aiming to pass a test and so on. Either you know the stuff or you don't. So I was lucky, and by chance, later on, I learned that I had the highest score in Bath on that particular test, the 11 plus test for mathematics. Now, at age 11, I'd already set my mind on several things. Prior to that, I wanted to be a mathematician. I thought I'd be a professional mathematician. And then I read this book that was called How I Became a Detective, a traditional police detective. And after I finished that book, I knew this was what I wanted to do with my life. Changed me completely in terms of what I thought about. And then my father bought me a chemistry set. And I discovered that with this chemistry set, this was in the old days when chemistry sets were really good. They weren't ones where, all the experiments were boring. I discovered you could make fireworks and you could make explosives with this set. And I used to go down to the library and pull up books and read about this and sent my father off to buy chemicals that I needed. And that set me on the trail for chemistry. I was going to be a chemist. And so you see, even by the time I was 12 or 13, I'd already switched my fields several times from mathematics to a detective to becoming a chemist. And I think this is something I always tell people, young people, they should do. Don't just think that the thing that maybe your mum and dad said, "Oh, this is what you've got to do with your life," or the thing you think, "This is what I want to do with your life." Find out about other things, read about other things. Listen to lectures about other things, because you just never know when you're going to come across something that is more interesting and which you can be more passionate about than what you're already doing. So I think this is incredibly important. Now, when I went to grammar school, I had a terrific math teacher. He was really good, and I loved mathematics, but I knew chemistry was what I really wanted to do. I learned to play the violin. I loved that. I thought that was great. I lived close to a limestone escarpment with lots of caves, and I used to go into caves, and I used to find bugs in the caves, and we would send them off to a collector. I also discovered jazz. I loved jazz as well as classical music, so that was good. But then something interesting happened when I was, I think, 17. In the sixth form of the school that I went to when you were 16, you spent that year getting ready to learn to pass the advanced level exams, which would tell you whether you could go to university or not. And, you know, I love this. I love learning. I've always liked learning. I like finding out about new things. And so I took this first year, and then the second year, instead of learning new stuff, they just kept going over this same old stuff. And I got bored. And so I stopped going to school. And I used to go in the morning, sign in, and then I'd go and play snooker, billiards and snooker, down at the local YMCA. And the teacher found out about this, and they threatened to expel me. I thought I was going to be thrown out of school. But the thing I did best at the time was to play snooker, and also billiards. I was pretty good at billiards. At snooker, though, I was the West of England junior champion. And so when I thought I was going to get thrown out of school, I went to audition to be the sparring partner for the world champion at the time, a guy called Joe Davis. He was an Englishman, and his sparring partner had retired, and he was looking for someone to go on exhibition matches with him. And so I applied for the job, and I thought I was going to be pretty good. But then the headmaster said, "Oh, no, we'll let you come back to school after that. We're not going to expel you." So I always thought I missed a big opportunity to become famous there, because I was really good at snooker and I loved it. But actually, the most important lesson I learned at that time was I was playing in a tournament, and I was trying to pot a ball, and I didn't pot the ball that I was trying to, but I potted another one instead, and so I could continue the break. And the next shot, I was careless and I missed it. And an old man, I was 17, 18 at the time, an old man came up to me, must have been 80, and he said, "You know, you've got to learn one thing, that when luck strikes, you have to take advantage of it. You've got to try twice as hard on the next shot. Whatever it is, if you have a lucky break, try twice as hard to take advantage of it. You're playing tennis. The ball just hits the net and drops over. Your opponent can't get it. You've got to concentrate twice as hard on the next shot, because otherwise, why did you have the luck? Everybody has luck. You look back in your lives and say, well, where did luck take me one way instead of the other? You'll be surprised at how often luck has charted your path. And then you can ask yourself, and did I always take advantage of it? When you're young, learn this lesson, take advantage of it when it comes your way. Now, as a result of this messing about and not going to school in this second year, and I didn't like physics very much. We had a terrible teacher. And so I failed physics at advanced level, and so that meant I couldn't go to Oxford or Cambridge because I just didn't have the scores for that. And so I had to stay on at school for an extra year, and then I applied to 2 universities that had good chemistry departments. One was Sheffield University, one was Southampton University. Southampton University turned me down, Sheffield accepted me, and so I ended up going to Sheffield instead. And that turned out to be really a very wonderful thing. I had a great time there. They had a very good students' union, so they had a good snooker table, and that was good. I learned to play bridge when I was there. I loved bridge. I loved card games. I also realized, thanks to a man who I did my graduate work with, Professor W.D. Ollis, that actually chemistry was about solving puzzles. Chemistry wasn't just learning the names of reactions, the names of the famous chemists. It was about solving puzzles. And so in a way, chemistry, and especially when I went to do research, was being a detective. that was great. So here was something I loved when I was young, and now here I am doing it, pursuing an academic thing. Now, during this time, I really ran into someone who made a tremendous difference in my life. His name is Kazu Kurosawa, and he was a postdoc in the lab where I was a graduate student, but he was the best teacher I've ever come across. He was just great. I would say, oh, you know, I was given a project, I'd do it, and he'd say, oh, you know, you should do it this way. This is the experiment you should do, and this is why you should do it. And then if I had trouble with the results, he'd explain how to interpret the results, and if they went wrong, why they went wrong. And he was just on top of everything, and he was great. And he also taught me how to play Go, this game on the bottom left, which for many years I was never beaten by anybody who was not Japanese. Unfortunately, after 1 year, he left to go back to Japan. We used to play Go by mail. Can you believe this? We would play this game by mail for probably 3 or 4 years after he went back to Japan. But I got all of the results I needed for my thesis in 1 year. At the end of year 1, I could have written my PhD thesis, but in England, you have to do it for 3 years. You know, they won't let you get away with 1 year. And so after the second year, during the second year, I sort of did some things that I thought might be interesting, but Kazu had gone, and there was no one there really to advise me what to do and tell me what happened when things went wrong. And so I got some results, but I didn't need them because I already had everything for my thesis. And then in the third year, I started going over to the library and reading about things outside of my subject. And I read this book called The Thread of Life by John Kendrew. John Kendrew was a Nobel Prize winner. He won it for determining the first structure of a protein. And this book was all about the origins of molecular biology, how molecular biology became a discipline in its own right. And by the time I finished that book, I knew this was what I wanted to do with my life. I wanted to be a molecular biologist. And so I went and talked to Professor Ollis, and he said, oh, there's no such subject. And I talked to some of the other chemists. You know, you've got to remember, this was back in 1968, a long time ago. Everybody else on the chemistry faculty at Sheffield all said, there's no such thing as molecular biology, do chemistry. You know, this is what you've been trained for. But there was one young assistant professor called Mike Blackburn, and he said he believed there was a molecular biology subject. It was a good one, and he encouraged me to go for it. So I applied for 6 postdoctoral positions at labs where they were organic chemists who really were doing things that had a very big biological bent. And I applied to 6 of them. 5 of them turned me down, and 1 accepted me. And the one that accepted me was this man, Jack Strominger. And he, at the time, was a professor in Wisconsin, at Madison, Wisconsin. And so when he accepted me, I thought I would be going to Wisconsin. But then 2 months before I had to pack up and get my family together, I had a wife and 2 children at the time, before I had to pack up and move to Madison, he said, oh, can you just delay for a month? Because I'm going to Harvard. I've just been appointed a full professor at Harvard, which was his lifelong ambition. And so when I got there, instead of going to Madison, I went to Cambridge, I went to Harvard, and had a great time there. But one of the reasons I had such a good time was because Jack, who was well known to be a real taskmaster, he would make sure people in his lab worked hard and did the projects he wanted. He had just achieved his lifelong ambition. He always wanted to be a professor at Harvard. And he put me to work with another postdoc and sort of finish up a project that was getting started, but he paid no interest in it himself particularly. You know, if you got something interesting, he wanted to know, but he wasn't really into this. He was not on top of me all the time. And the postdoc I picked the project up from was Australian. He went back to Australia, and I was left by myself to do this project. Now, the postdoc had been trying to sequence a tRNA. There was a transfer RNA in Staphylococcus aureus and Staphylococcus epidermidis, that worked for bacterial cell wall synthesis. This was the way in which the glycine, the little chain, 5 glycines that formed the outer part of the peptidoglycan, a tRNA was produced. And that tRNA only worked in bacterial cell wall biosynthesis. It didn't work in protein synthesis. And so the question was, what is funny about it? What do you need to do? And so the essence was to sequence this tRNA and see how it differed from all the others. Now, at the time, there was a man called Robert Holly who had won a prize, the Nobel Prize, for figuring out how to sequence tRNA, but he was using a very old-fashioned method. And in the meantime, Fred Sanger, the gentleman underneath the red arrow, was setting up a new method for sequencing RNA. It used p32 labeled RNA, and it was a really cool method. And I'd read about it in the literature. And so I said, well, I'm not going to do it by this old-fashioned method. I'm going to try it with Fred's method. And I wrote to him and said, could I come to your lab, work in your lab, and learn how to do it? And he wrote back and he said, sure. And so I went, I spent 6 weeks in his lab. He is my ideal of what a scientist should be. He'd already won 1 Nobel Prize at the time for determining methods for sequencing proteins. Later, he won the Nobel Prize for determining how to sequence DNA. And in the meantime, he did RNA. He should have had a 3rd prize for sequencing RNA because it was a massive achievement. Anyway, spent 6 weeks in his lab. The day I arrived was a Sunday. And I went into the lab and, you know, there was a guy there busy washing dishes and so on. And I said, oh, you know, I'm Rich Roberts. I've just come to work with Fred Sanger. And he looked up and he said, oh, I'm Fred Sanger. Hi. This was a man. He'd already won 1 Nobel Prize, developed a method for sequencing RNA, and here he is washing the dishes in his lab. And this was Fred Sanger. He was just a very, very modest man. He was terrific, just a terrific scientist. And he was one of these people who, if you'd done something really stupid, instead of saying, oh, you know, that was stupid, you shouldn't have done that, he'd look and he'd say, yes, interesting you did that. Did you think about doing this? And that was as much of a reproach as you ever got from him. He was just a very polite, very caring person. And he didn't want to offend anybody. Great guy, really a great guy. He shared the prize in 1980 with Wally Gilbert. And Wally, if anything, had set sequencing back because the method he came up for sequencing DNA was not very good. And it violated Fred's principles of what you needed to do when you were determining sequence. Fred should have had the prize by himself. He didn't need to share it with anyone. Now, after I finished my time at Harvard, I was looking for a job. I applied for a job in Edinburgh, and I'd been told that by a professor there, that this was the perfect job for me, that, you know, all I needed to do was apply and I would go. And so I sent in my application, and then I never heard back. And then I got an invitation to go for a job at Harvard and was offered a job in the biology labs at Harvard. And then I was talking to a man called Mark Potashny, who was a well-known biologist at the time who worked with Jim Watson. And he came up to me after a seminar one day and he said, you know, Jim would like to hire you to go to Cold Spring Harbor. And he said he's going to come talk to you about this. So, you know, I waited and waited. I'd never met Jim before then. And after a couple of weeks, Mark says, well, Jim actually doesn't know who you are. Perhaps you could go and introduce yourself. So I did. Walk into his office and say, you know what, I'm Rich Roberts. I heard you wanted to talk to me. And his first words were, yes, I want to offer you a job at Cold Spring Harbor. And we talked for a few minutes, but not really about that. And then I was just about to leave the office and he said, oh, you should probably come down to Cold Spring Harbor and give a talk, you know, just so that you see what the place is like. But don't worry, you got the job. The job is yours, he said. And so I did go down to Cold Spring Harbor and the job was mine. And I went down there and it turned out to be quite good. although I've got to say Jim Watson is one of the most difficult people I've ever run against. I worked there for 20 years. For the last 8 years, I was assistant director for research, which meant I had to interact with Jim on a very regular basis. And at the time, looking back on it, I would have done well to leave and go and do something else. But I'll tell you about that other thing later on. Oh, sorry. Now, before I went to Cold Spring Harbor, what Jim wanted me to do was to determine the sequence of SV40 virus. And what he wanted me to do was to use the methods that 2 other groups were already using, I discovered later on. One was Walter Fears in Belgium, and the other was Sherman Weissman over at Yale. And they were using basically Fred Sanger's method for sequencing SV40. And I thought, well, you know, I don't like doing the same thing that anybody else does. I always think if someone tells me, oh, you know, you should do that, I always think that must be pretty boring. I like to do things that I thought of. And so during, I think, 3 months before I went down to Cold Spring Harbor, I started to hear this talk by Dan Nathans, the man in the top left, who eventually won the Nobel Prize together with Werner Aber and Hamilton Smith. But he was talking about a newly discovered enzyme that recognized very specific sequences in DNA. And so you could take a big DNA and cut it into small pieces with this enzyme. And one of the reasons that Fred Sanger had first concentrated on RNA was because there were plenty of small RNAs to practice on and no small DNAs. You know, anytime you're going to start with a new method, you have to find something small to practice on, where maybe you can get the results in 2 or 3 different ways so that you know everything is working properly. And it occurred to me that I could perhaps use this enzyme to develop methods to sequence DNA, and especially if there were more of them, and I quickly learned that there were already 2 or 3 more known, that we could get a flock of these enzymes and use them to determine the sequence of DNA. And so as soon as I got to Cold Spring Harbor, I started working on this. Almost immediately, Jim Watson threatened to fire me because I wasn't doing the project that he thought of. But I managed to slip away from that for a while. And we started looking for new restriction enzymes. And pretty soon, we found almost everywhere we looked, there were new restriction enzymes, and they recognized different sequences. Of the first 100 restriction enzymes that were discovered, 70 of them were found in my lab, 70 out of the first 100. And these were the enzymes that really formed the basis of the biotech industry. Without these enzymes, cloning would not have taken off the way it was, the way it did. The biotech industry would have been slowed down considerably. So these were really important, these enzymes. And it occurred to me, people used to come to Cold Spring Harbor to meetings, and they heard that I had this collection of restriction enzymes, and they'd come down, they'd have a tube of DNA in their pocket, and they'd sneak out of the meeting, come down to my lab and say, hey, I've got some DNA here. Do you think you have a restriction enzyme that will cut it? And so we set up and we allowed them to do that. And then at the end of it, they said, oh, could you give me a little bit of that enzyme? You know, because they didn't want to have to make it themselves. And so we were giving all these enzymes away. Some people wanted the strains, we'd give the strains away. And it occurred to me that there was a business here. And so I went and talked to Jim and I said, look, why don't we start a business at Cold Spring Harbor making these restriction enzymes and selling them to the world, because researchers everywhere wanted them. Well, he says, no, he's not interested in that. 2 things. Didn't think you could make any money at that. And the other was that he thought it was rather dirty to be associated with industry. He didn't want anything to do with that. Fortunately, I talked to someone down at NIH. He put me in touch with a guy in Boston called Don Cohn, who had had a similar idea. He wanted to start a company, he'd been making other things, but not very profitable. But he was thinking of selling one of the first restriction enzymes. And so I called him up, talked to him, went up there, and that is how New England Biolabs really got started, and that's where I now work. I'll tell you a little more about that later on if time permits. Now, at Cold Spring Harbor, I had a very lucky encounter. I was going to a meeting in Belgium. I was on the train from the airport into Brussels and sat next to this gentleman, Norton Zinder. He was a professor at Rockefeller, and we got talking, and it turned out that he was very interested in restriction enzymes, thought that what I was doing was terrific, and he was a good friend of Jim Watson. Now, throughout this time, Jim was constantly threatening to fire me because I wasn't doing what he wanted me to do. And so I would call Norton, Norton would call Jim, and then Jim would back off and not fire me. So this man rescued me on so many occasions from Jim Watson that I really owe him a great debt. He was just terrific. And I think this is also something that's good advice for young people, that it's always good to find someone who has your back, who can help you when you really need help. Sorry. Now, at Cold Spring Harbor, while we were finding all these restriction enzymes, we were also interested in doing some other things. And I had this one postdoc who came, Richard Gelinas, and I'd met him at Harvard. He'd been a graduate student at Harvard, came to my lab, and we were looking for a nice project. And we got interested in adenovirus RNA. And in particular, we thought maybe it would be interesting to see whether a promoter, that is the region in the DNA that tells the RNA polymerase where to start making RNA that is going to go off to the ribosome and make protein. We thought it would be interesting to know if a eukaryotic virus, because adenovirus grows in human cells, whether the sequence there was the same as the one that you find in bacteria, because that had just been determined by Wally Gilbert, Mark Potashnick. And so we started on this. And if you look at the little diagram here, you'll see that there are some early messenger RNAs. They're shown by the little arrows, E1, E2, E3, E4. And what we thought was that if we could find exactly where that RNA started, what was the sequence at the start of the RNA, we would just have to look upstream on the DNA, and then we would know where the promoter was. Well, we tried to do that with early RNAs, and it failed miserably. We couldn't get enough of the RNA in order to do the experiment properly. So then we turned to the late portion. So late in infection, all of these transcripts, L1, L2, L3, L4, and so on, all get made. We knew approximately where the genes were that they were encoding for. And so we thought it would be good to just pick any one of these if we can get any 5' end and then look upstream and see what the promoter was. And so Richard did that experiment. While we were doing it, a big discovery was made, and that is that these RNAs have a special chemical structure at their 5' end. And thanks to that, I was able to come up with a very clever way of isolating these 5' ends. And it's basically shown here. You take the messenger RNA, cut it with ribonuclease T1 that cuts after every G in the RNA, and then just look and see what is at the 5' end. And we had a two-dimensional method of doing this. And we were expecting to see maybe 10, 15, 20 different 5' ends. And you can see that on the left, there were just really two major spots. It turned out that one of those spots was related to the other in that a phosphate had cyclized on it. And really, there was just one. And this meant that all of these late messenger RNAs all had exactly the same 5' end. And so we spent a lot of time trying to figure out how this was possible. In the meantime, we published this paper about it. But then we wondered, well, maybe what is going on is that these RNAs are actually derived from something much longer. And this was something called an R-loop experiment. The little red blob is the cap, this funny chemical structure I showed you. We already had some evidence that the RNA, the full length of the RNA, was not coded in just one place on the adenovirus genome. To cut a long story short, we did something called an R-loop experiment, and we discovered that what was going on was that this piece of RNA was composed of actually 3 parts at least, shown in the bottom right-hand corner. And this was done by an electron microscopy experiment that I devised while Richard and I were talking one Saturday afternoon. And we had an EM lab that was very close to us, Tom Broker and Louise Chow. Louise is really good. She was a wonderful electron microscopist. And we went down to them and we said, you know, I have this idea for an experiment. Could you do it? And she said, well, you know, it's not been done before, but yeah, we could probably try it. And so on the Sunday, Richard made the reagents that were needed to do the experiment, finished it up on Monday, and on the Tuesday morning, we took the RNA and the DNA down to Tom and Louise, and the very first molecule they looked at in the electron microscope just looked exactly like the one I'd drawn up on the board. And they started to look further, and everyone they looked at, they found this same structure. And now normally when you do electron microscopy experiments and get a result, because you're only looking at a very tiny proportion of all the molecules, you have to do a lot of work to show that what you see is genuine, that all of the molecules are like that. But it turned out we'd already spent a year, Richard and I, doing all of these other experiments, and this was just the icing on the cake. So this result got on a Tuesday morning. On Tuesday afternoon, people were talking about Nobel Prizes. We went around talking, telling everybody we could think of about how great this was. A lot of other labs, it turned out, had data that could be explained now that they'd never been able to explain before. And so this turned out to be quite a big deal. And this is the paper that we wrote about it. It's called An Amazing Sequence Arrangement of the 5' ends of adenovirus 2 messenger RNA. There's another little lesson here. The reviewers of the paper didn't like the word amazing in the title. They said this is not appropriate in a scientific paper. All three reviewers said this. And Ben Lewin wrote back, he was the editor, wrote back and said, well, you know, just find another word for this. Great thing, but find another word. And so I called him up and was talking about some of the other points that the reviewers had made. And during it, I said, hey, when you first heard about this, what did you think? Oh, it was amazing, he said. I said, I rest my case. And so that's how the word amazing got in. As far as I know, that was the first time it was ever used in the title of a scientific paper. Now, in 1993, I won the Nobel Prize with Phil Sharp. He had made a similar discovery at MIT quite independently of us. And so we went to Sweden, had a great time. I tell you, the Swedes, they really know how to throw a party. Unbelievable. We were there for 10 days. Every single day, there would be something would happen at lunch, something would happen at the evening. And if either of those was the only thing that happened to you in the whole year, it would have been the highlight of the year. Imagine that for 10 days, every lunch, every day. Very difficult to stay sober, I can tell you that. Now, one of the things you have to do when you go to Sweden for the Nobel Prize is give a talk. And so the talk that most people give says all the wonderful work they've done since they've made the discovery. Well, I hadn't actually worked in that field for quite a long time. We tried to for a while, but got delayed. But just before I had to go to Stockholm, we made this discovery. And this was about a DNA methyltransferase. So this is an enzyme that puts a methyl group, CH3, onto DNA at specific sites. We've been trying for about 3 years to get a crystal structure, and then we got this structure and discovered when this enzyme binds to DNA, it takes the base that it's going to put the methyl group on and flips it right out of the helix into the enzyme, puts the methyl group on, and then it goes back. And so here I had a wonderful discovery that I could talk about for my Nobel lecture. You know, and if you want to make a discovery and tell someone about it, no one better than the Nobel Committee. I'm still waiting for the results of that, though. Now, I just want to spend a couple of minutes talking about New England Biolabs. So New England Biolabs we're just celebrating our 50th. It was actually 75, early 75 when it started. We're celebrating our anniversary. Don Combe had been a professor, an associate professor rather, at Harvard Medical School in one of those departments where they never give tenure. Got fed up with the system. He says, I'm going to go start a company, going to make some money, and I'm going to use the money to support research, basic research, and I'm going to become NIH and Harvard all rolled into one. And when he died, he thought that that's what he'd done. We now have 100 people at New England Biolabs, all doing basic research, all paid for by the company. No NIH grants, no involvement of NIH, just working on things that they think are important. Now, in 1992, I got fed up at Cold Spring Harbor and was looking for something else to do. I'd thought about maybe starting a DNA sequencing company, but Don said, you know, well, why don't you come up to Biolabs? You know, you've been our chief scientific advisor since 1975. Please come up and be our research director. So he basically made me an offer that I couldn't refuse. So that's where I went. This was the very first catalog of restriction enzymes. I'd taken already all the restriction enzymes, given them to them. In the first year, back in 1975, they made $200,000 selling restriction enzymes. 200, that's a lot of money back in 75. We've continued to do pretty well. We've just moved a few years ago, 2005, into this new building. We've just put a big extension on that. We have about 700, 750 people working worldwide, most of them distributors, but in Ipswich, our headquarters, we have about 370 people, 100 just doing basic research. This is the company Don, this is the current CEO. His name is Sal Russello, and he's a very good guy, very nice guy. Now, I've always loved puzzles and games, so one of the things I did with my Nobel Prize money was to put a croquet lawn in front of my house at quite considerable expense, but it's one of the best uses that I've ever made of the money that came my way. I love croquet. I learned to play it when I was at Cold Spring at the MRC lab in Cambridge in Fred Sanger's lab. I've had a bunch of honors. I was knighted by then Prince Charles, now King Charles. I met the Queen on a number of occasions. She was fantastic. I've also helped organize the Nobel laureates to do a lot of good work, one of which was to get some Bulgarian nurses out of jail in Libya. They've been accused of spreading HIV in the children's hospital and put on death row. I got the Nobel laureates together. I went over to Tripoli. I talked to Gaddafi's son. And at the end of the conversation, he said, we're going to let them go. So I discovered that having a Nobel Prize gives me a little bit of an edge here and there. Now, if you'll excuse me, I'll just spend a little bit time talking about luck. So billiards, of course, was good. Kazu Kurosawa, totally luck. Harvard, not Wisconsin, totally luck. Looking for eukaryotic promoters in adenovirus, we did an experiment and it failed. And I think this tells you something very important. Failure is really the best learning vehicle out there. When you do an experiment and it fails, you must do your best to find out why it failed, because this is when you learn. Probably one of the luckiest things that happened to me was taking an early plane. You probably heard about the plane that went into the World Trade Tower in 2001, demolished it. I was booked on that plane until 2 weeks before it took off. I was going to a meeting in California, and the meeting got moved forward by 1 day. And so instead of September 11th, September 10th, I was over in California, flew over on this plane, arrived over there, put the TV on in the morning, and see the plane that I had been booked on going into the World Trade Tower. That's pretty good. The Unabomber. This was a guy, he was actually a mathematician from Harvard, from MIT rather, and he went off the wall and he started sending bombs around to people. He sent his very final act, his final batch of letters and so on. He sent 5 letters to 5 people, and one of them had a bomb. I got one of the letters, but not the bomb. The bomb went to someone in San Francisco. And then at the bottom, I talk about studying bacterial RM systems. I did it because I wanted to use them to learn how to sequence DNA, but in fact, they turned out to be far more useful than for sequencing DNA, and the rest of the world figured out what to do with them. Now, I want to finish by talking about something that I've been drawn into very recently, and that is to be concerned about the way that we men tend to treat women in science. It's really been quite alarming. I've read these 3 books. The Exceptions is about Nancy Hopkins, who detailed what happened to her at MIT. Breaking Through is by last year's winner, Kathleen Carrico, the difficulties that she faced as a woman, being a scientist. And then finally, this book, Invisible Women, which came out a few years ago, but it specifically talks about the data showing how society, for many things, and healthcare is a very good example, where it's biased against women and towards men. And I think a lot of us men, and I'm guilty as anybody on this, we often think if we treat women as though they're men and we try to treat them equally, that that's good enough. But it's not good enough. Women are not men. Women are different. They have different problems. Childcare is a problem. Menstrual periods can be a problem. They have all sorts of things that differentiate them from us. And I've started to realize after reading these books and also this movie, Picture a Scientist, this is really an important issue. And I make a point now, if I'm going to be invited to go and talk somewhere, I want to make sure that there are women talking on the stage, not just men. I want to make sure I won't go. If I get invited and I hear there are no women invited to go, I won't go. If there's 1 woman and 10 men, I won't go. I want to see an equal number there. And I've also talked to a few of my Nobel friends about that, and they are taking this same attitude because this discrimination against women has to stop. So I will stop at that point. I thank you for listening, and if you have any questions, I'm happy to take them.